Alright, we're going to talk about cellular respiration starting with glycolysis. So glycolysis is basically you take your glucose molecule and through the input of 2 ATP molecules you get 2 pyruvate molecules. Which are basically half of this glucose. 1/2 glucose. Because this has 6 carbons and this has 3 carbons apiece. To show you what this looks like, over here we go. You start with this. You start with your glucose molecule. Through the input of ADP or ATP, you take the phosphate group of that ATP and put it on the glucose to create a glucose 6-phosphate. And then you do this step again to have the 2 phosphates on here, that when it splits, you're left with the 2 pyruvate molecules, as you can see here. So now you have your 2 3-carbon molecules, and now with each pyruvate, which are the 3 carbons, hydrogen, oxygen, and a phosphate, you, with the addition of an NAD plus and another phosphate, you create a 2-phosphate and 3-carbon pyruvate. That gives you NADH and a positive hydrogen, which is just a proton. Now we remove one of the phosphates to create ATP. So we have the 2 phosphates on this. We take it off, it rearranges it, and it gives us an ATP back from one of the 2 that we gave. Now this remaining molecule rearranges itself, kind of flipping itself. It flips the oxygen and hydrogen with the phosphate group. Now the same thing occurs where we take ADP, take this phosphate off, and that creates ATP, giving us pyruvate again. So now that we have our pyruvate, these can go into the mitochondria to begin the next step, link reactions.
So after glycolysis, it moves into the mitochondria, and you have pyruvate oxidation. Pretty much it's self-explanatory. The pyruvate is just oxidized, which form 2-acetyl-CoA, and this then produces 2 NADH that will go through a electron transport carrier into oxidative phosphorylation, which we will talk about later.
Alright, so once we have our acetyl-CoA, this goes into the citric acid cycle. Citric acid. We got our citric acid cycle with our acetyl-CoA. Going in, and then through a process of redox reactions, we begin to get start our cycle and we get 2 CO2. Well, I'm just going through the products of this. We enter 3 NAD plus, so the more neutral, stable NAD atoms. And we get out 3 NADHs, and also we get 3 protons, or 8, which are H pluses. As the cycle continues on, we enter again the same type of thing where we go ADP plus that initial phosphate, that third phosphate, and then we use the energy gathered from the NADH to give us an ATP molecule. So we keep going on. We put in a low energy FADH, and we get in high energy FADH2. So we lose 2 hydrogens in that product. And then that creates the cycle for the citric acid cycle. So with acetyl-CoA, we end up with 2 carbon dioxide molecules, 3 NADH, 3 protons, so 2 CO2, 3 NADH, that's the high energy one, 3 positive hydrogen, which is just a proton. We make a molecule of ATP by binding an ADP and a phosphate, and we get an FADH2. All of these, maybe not the carbon dioxide, but these go into the next step of oxidative phosphorylation.
Okay, so that brings us to our last stage, oxidative phosphorylation. Okay, so NADH and FADH2 shuttle high energy electrons into the extracted from your food and gone through all those cycles. Yeah, okay, we got it. Into the electron transport chain, built into the inner membrane of the mitochondria, that is where oxidative phosphorylation occurs. And so for oxidative phosphorylation, 6 oxygen molecules that you extract from breathing, and all 10 NADH, which is from the previous cycles, all go in to this cycle. And that produces 26 to 28 ATPs, which is the most amount that cellular respiration, or the most amount out of each cycle produces. And it varies because of how the shuttle of transport electrons from NADH travel. And yeah, Andrew's gonna step in.
So, to create these ATP, you have to take the hydrogen. It's a you pump positive hydrogen. You take hydrogen positive hydrogen ions and using the electron transport chain within the cristae, you take these and you pump them out of the mitochondria. To create a gradient, an ion gradient, that then that's what releases the energy for the ATP. And then with chemiosis, chemiosmosis, sorry. You take that gradient that you've created by using energy, and you allow those positive hydrogen ions back into the mitochondria up here. So they leave using energy and then they rush back in with a different set of molecules embedded in the membrane that release enough energy to synthesize ATP. So when this comes back in the mitochondria, you use the free energy to synthesize ATP. And that's where you get your 26 to 28 ATP molecules in this stage, varying on solute concentrations and environmental factors like temperature or different things like that. So that oxidative phosphorylation has to do both with the electron transport chain out of the or through the cristae, and chemiosmosis, allowing the ions back in, synthesizing ATP in the process. Thanks.